Uh, Today, we shall start an interesting topic, I think, which is very important for bioactive natural products, your course BTL530. That is, how can we discover or isolate uh, bioactive natural products from the natural sources? Let us show you some of the slides. Is it visible to you?
Yes, sir.
Okay. Today we shall discuss on analytical methods for discovering bioactive natural products. Bioactive natural products, you know, they are present in various sources, terrestrial and marine organisms like plants, bacteria, algae, fungi, sponge, even in sea invertebrate organism. Uh, Even some vertebrate organism also. And these compounds are basically synthesized from the primary metabolites, and they are basically, most of them are secondary metabolites. Some of them are also, protein or peptide-like substances, and they are derived from the primary metabolites and biosynthesized in the plants, in the cells of the organism for their own defense, for their own fitness, for their own communication, and protect themselves from different biotic and abiotic stresses. So these are basically organism synthesize them in their cell for their own purpose. But a human being, due to the intellectual ability, they discover the materials and then use for their own purposes, for example, drug or agrochemicals or like that. So it is very important how can we discover them. You know, natural product compound obtained from and synthesized from the living organism, they are called natural product. And chemistry is the study of matter and energy and the interactions between them. Already, you know this fundamental definition. So we can define natural product chemistry as the branch of chemistry which deals with the isolation, identification, structure elucidation, and study of chemical characteristics of chemical substances produced by the living organism. That means natural products are uh, some chemical substances, and the subject deals with the natural products are called natural product chemistry. And natural product chemistry is involved in isolation. That means compounds in the organisms in their cells remain with other, compounds. So mixture of compounds. So you have to isolate your target compound, then identify it and elucidate the structure, chemical structure, and then check the biological activity. And even you can synthesize, prepare it in the laboratory. So by natural product chemistry is a very important branch of science. And also it is a branch of chemistry. The field of natural product chemistry is one of the most promising in the continued search for new pharmaceutical agents. Here is the analytical process. I'm giving you an example of discover how we can discover bioactive natural product from plants. But a similar process could be applied for discovering the natural product from other sources. In case of plant, you know, bioactive natural products or secondary metabolites can be present in the root, stem, heartwood, bark, leaves, flowers, even in any organs of the plant. For example, the bark, some of the plant bark, tree bark, they are very, rich in secondary metabolites to protect them from the insect and other herbivore attack. So if you target to discover compound from the plant bark, what you can do, you can collect the bark from the plant and then uh, allow to dry the bark in the shade, not in the sunlight, or not using any heat, because high temperature or sunlight can cause decomposition of some of the secondary metabolites. Some metabolites are sensitive to light, some metabolites are sensitive to uh, temperature. So if you expose them in these two abiotic factors, then you may lose your target compound. So after drying, why we dry? Because water is a problem. During isolation process, water sometimes a problem, but you can also uh, grind, or you can also extract the bioactive compounds from the fresh bark also. But anyway, uh, uh, but in that case, you have to crush the uh, uh, bark, uh, so that the molecules are present in the cell, so you can damage the cell. And then as I mentioned, you can grind. Grinding is just to expose the damage the cell, so that organic solvent can enter into the cell interior and dissolve the organic compounds, that is bioactive compounds, in the solvent. And solvent we usually use uh, you know uh, non-polar solvent, but you can use even water. But water, handling of water during evaporation is really difficult. But anyway, if you use acetone, if you use ethanol, if you use methanol, or you can use dichloromethane, uh, depending on the target of your compound. If it is highly polar compound, then you need to use polar solvent. For example, ethanol. Ethanol is highly polar, just above the water, lower the water, and then methanol, and then you can use even chloroform. But chloroform, as it contains chlorine, not good for the environment. Better is dichloromethane, diethyl ether, or N-hexane. N-hexane is the least polar compound. So oily compound, diethyl ether, that means petroleum ether, or N-hexane, can be used to extract the oily compound. Those are hydrophobic or very low polar. But if you use ethyl acetate is a middle even a polar compound, a non-polar compound can be extracted by using ethyl acetate. And ethyl acetate, acetone, even you can use, in case of acetone, 60% acetone, that means 40% water. If you target the polar compound and get large range of non-polar to polar compound. Anyway, after grinding, you can put the powder or paste of the materials in the flask, and then you can use the organic solvent, as I mentioned, and shake occasionally. And you have to close the cap of the flask very tightly, so that organic solvent, most of them are less polar, and can be evaporated and create a fire and others. So you have to be very, very careful when you extract the compound, because they can evaporate very fast. So if you put 3 or 4 days and occasionally shake, then the organic solvent can penetrate the cell interior and extract, dissolve the organic compounds, that is bioactive compounds or non-bioactive compounds, all you will get in the solvent. But sometimes we use 3 times extraction. First time, what you do, you just filter the solvent and get the residue, and then again add the solvent and collect the solution that is the solvent with the chemical compounds. After getting that, you need to evaporate the organic solvent. So you collect the extract and then evaporate them by using vacuum evaporator. We have a vacuum evaporator, so organic solvent can be evaporated, and you will get a gummy or dried materials which contains lots of organic compounds as you use the organic solvent. Because in chemistry it is called like dissolve like. That means organic solvent can dissolve organic compounds. So you have now mixture of compounds, and mixture of compounds using a small amount of how to say, you can check the a mixture by using TLC. TLC, that means thin layer chromatography, and using different solvent mixture, and you can check how many compounds present in the mixture, and measure compounds. You can easily get the spot, because if you put in the TLC tank a TLC plate and small amount of mixture of compound dissolved in organic solvent, then after few minutes or 30 minutes, you can find this type of separation. And you can use several times different combination of solvents so that they are separated well, because same mixture of solvent you can use in the column chromatography. So in the column chromatography, you can use silica gel or Cosmosil or some other adsorbent. You can pack the column like this way, and then your extract in small quantity of solvent which can dissolve the compound. You can add here, and then the solvent system you discovered by the TLC, you can add the mixture of solvent organic compound, organic solvent, and here you can the molecule will pass through the column, and column has a different column packing materials has different properties. Some of the column is just allowing heavier molecule to pass through faster, or some of the column, depending on the solvent system, allow you to pass through the a high polar compound fast or low polar compound fast, depending on the organic solvent as well as the column. So that means the mixture, in the mixture, if you put here and if you use appropriate solvent system, when they pass through the column, due to the adsorbent capacity of the column packing materials, depending on the molecular size and polarity, compound pass through in different speed. As a result, they will be separated. So you collect the 10 ml per tube and check them in the TLC plate. And you can find maybe 3 tubes, same compound or like that, and you can get this type of separation. This is the main mixture. And when you get the pure compound, you may need to do several, column chromatography. After getting semi-purified 3 or 4 compounds, then you can do second time, and finally you can get the pure compound in the TLC. And TLC will give you quick result that whether your compound is pure or not. You can change the solvent system and see whether this spot contains single compound or different mixture of the compounds. And after having a pure compound, then you concentrate, evaporate the solvent and get the pure compound. And after having the pure compound, you go for structure elucidation. How can you do? You need to do mass spectrometry. That is, first you need to know the molecular weight of the compound and also chemical composition, carbon, hydrogen, oxygen, and other elements in which ratio they are present. For example, glucose C6H12O6 like this. So molecular formula and molecular weight you can get from different kind of mass spectrometry. For example, high resolution mass spectrometry, or EI mass, or electric ionization mass spectrometry, or there are different kinds of mass spectrometry you can get the information about mass spectrum. And then NMR is a powerful tool, a nuclear magnetic resonance. This is a very powerful tool which will give you the information about the structure of the compound. NMR used to know mainly the carbon and proton, that is all organic compound contains carbon, hydrogen, oxygen. So carbon, proton, and oxygen, you can get information by using NMR data. So NMR, without any loss of the compound in the electromagnetic field, you can have the NMR data, and NMR data, how many carbons are present, and in carbon, for example, CH2, CO, or CH3, CH, how many each carbon, type of carbon present in the structure. And also you can get the information of the proton. And there are some other options also, CD, circular dichroism, and other spectroscopic method can be used. So these are the methods used to elucidate the structure. But in case of NMR data and mass spectrum data, if you get, you can put in the database and check whether your compound is known or not. If it is known, then the job is over. You can go for the biological study. But if it is unknown, then you need to precisely elucidate the new structure of the compound. Sometimes it is needed to do the synthesis, same compound. Once you elucidate the structure, you can prepare in the laboratory. There are some, in organic chemistry, lots of examples of synthesis of different kinds of compound. There are some methods. So you can synthesize, prepare the molecule, and then do the synthetic one and natural one in the same condition, same spectroscopic data. It should be identical, and biological activity also should be identical. Then the structure is 100% confirmed. So this is the way. And sometimes we use GC or HPLC because GC, gas chromatography, in case of volatile compound, very easy, and gas chromatogram peak can easily, if it is known, then can easily get the information about the structure of the compound if it is known compound. And HPLC, high pressure liquid chromatography, is also used for purification of the compound. But in HPLC and, for example, the column chromatography, there are differences. HPLC, small amount of compound, if only one or two impurities are there, then you can separate by HPLC. Small quantity, milligram level, you can separate. But in case of column chromatography, 100 milligrams or several hundred milligrams of compound, you can separate and get the pure compound. So these are the process. And after having the purified compound, when you elucidated the structure, then you go for checking the biological activity in the laboratory. There are several biological bioassay method. Bioassay method means testing your compound in the biological system to know its function. For example, whether it is antibacterial, or whether it is antifungal, or whether it is anti-cancer. We can use the compound very small quantity in the testing process to know its biological activity. And biological activity in case of natural product chemistry, if it is in micromolar concentration, it is active, then we consider it as a promising candidate for drug or any agrochemicals or other biologically active molecules. But if it is active at millimolar concentration or molar concentration, then it is not so interesting. But anyway, Once you get a compound with high biological activity and a new compound, in that case, usually natural product chemist synthesize the compound and derivatizes. That derivatize means changing the different side chain of the molecule to get the higher active compound, because natural ones sometimes not so strongly active. But if you structurally a little change, maybe it binds with the receptor more strongly and show the higher activity. These are the all about the analytical process. As I mentioned, mass spectrometry, here you can see FD mass, EI mass, a high resolution mass, multi-top mass spectrometry, metric assisted laser desorption, ionization, time of flight mass spectrometry, LCMS also. There is another way. In case of small amount of compound, you can use this machine, and machine will give you the mass spectrum as well as and the ionization pattern, HPLC, MC, LCM, SMS, NMR, I told you, carbon NMR, phosphate NMR, 2D NMR, or proton NMR. This is called proton NMR, not phosphate NMR. 2D NMR also another, GC, IR, CD, UV spectrometry. These are the methods called spectrometric methods used to elucidate the structure, as I discussed earlier. So thank you very much for listening the talk, and now it is your time. You can ask some question, and by the end of this week you shall have the midterm exam, because now the UGC withdraw their embargo on examination, so you can have the examination very I can take your examination very easily. So now the floor is open for your question.
Yes, sir. I have a questions. Like when I visited the Meherpur, I found that two adjacent wheat plant, one is severely affected with the fungus, while the other is not. So is it possible that the adjacent plant, which is not affected, maybe produce some antifungal compounds? So can we just extract compounds from there and use it for wheat plus disease?
Yes, for example, you have 5 varieties, and for particular disease you found one variety is immune, because plant has also immune system, immune against the particular disease. In that case, there is a high chance to get the secondary metabolites, which are involved in such immunity. Actually, immunity is regulated by the genes, and metabolites are the product of gene product, because when gene is expressed, then you can get even single metabolite or several metabolites may jointly involved in the protection of the plant from the particular fungus or like that. So it is a good idea to try, if you find a very good material which is very powerful in protecting the which is phenotypically very strong against a particular organ pest, then there is a chance of getting the uh, inhibitory or protecting compounds by using the analytical methods and spectroscopic methods. That is, you can target natural products.
Yeah, thank you, sir. I got my answer.
Thank you so much. And usually, crop plants, some of the wild relatives, they are very powerful in biosynthesizing the antimicrobial or anti-pest or anti, any stress molecules, because the wild plants are highly resistant. We are not taking care of them, and they are the reservoir of many valuable agrochemically important molecules. Even mushroom or other microorganism like bacteria, fungi, they might be involved, because sometimes we see some of the plants are highly immune against particular disease. In that case, uh, maybe not the plant metabolites, but the endophytes, that means microorganism inhabiting in the plant, they are protecting the plant from the enemy. So in that case, we need to isolate that microorganism, maybe fungi or bacteria or other, they are involved in producing such a metabolite that protect the host from the their enemy. So there are lots of strategies to discover the bioactive natural products from the nature. Any other question?
Uh, no, sir.
Thank you very much. Then wish you good luck, and I shall send you the slides today, and please go through the slide, and you shall have midterm one by the end of even weekend, no problem. I shall give you open book question. So I shall tell you which chapter you need to cover, and then sit for the examination, because we need to complete all of your evaluation. And when is the last date of this term?
Sir, July last week or August 1st.
Oh,
Probably sir, July
okay,
28.
very good. Then we have a good time, because I took a long pause because at that time there was an embargo for the examination. And if there is no examination, no evaluation system, usually students, they face problem of concentrating on the topic. Thank you very much.
Assalamu alaikum, sir.